ZS is a global management consulting firm with a deep focus in life sciences and healthcare. And at ZS, I lead up our AI practice specifically focused on pharma research and development. And so for today, I'd like to be talking to you about how we can unlock the productivity promise in the context of pharma R&D across the value chain, and share with you some of our experiences and learnings to deliver on this exciting and important new capability with AI. So I'd like to begin by talking a little bit about the productivity of pharma R&D. And now this is a topic of intense research over the past few years. And one of the phenomena that some of you may have heard of is called Eroom's law. It was written about in a paper in Nature back in 2012, where the authors looked at the productivity of pharma R&D in terms of the number of new molecular entities that pharma has been able to bring to market over the last 60 years. And what the authors observed is the fact that pharma R&D productivity has in fact exponentially declined, going from the 1950s all the way up to 2010. So that's what you see in the graph with the line in blue ranging up to 2010. Well, last year, this study was refreshed again in a new publication in Nature, where pharma R&D productivity was looked at over the last 10 years. Now, pharma R&D productivity is really important because pharma spends about $83 billion a year in R&D. This represents 25% of all of its revenues. And as an industry, pharma's investment in R&D is far ahead of any other industry. The next industry in terms of investment into R&D is in electronics and software, and they spend only about 15% of their revenue. So pharma invests a lot in the R&D process, of course, clearly for a variety of good reasons. And so given the scale of investment, it's really important to understand what have been some of the factors that have enabled pharma to reverse or break this Eroom's law phenomenon that have been observed in the past. And so what can we learn from these improvements over the past decade that are seen in this line in pink, where the pharma R&D cost of bringing a drug to market has improved from around about $2.5 billion back in 2010 to what's looking to closer like between $1 and $2 billion for every drug that has been brought to market. So some of the factors that have influenced this turnaround is that the authors note have been better information. And basically, this refers to just us having a better understanding of disease biologies, leveraging, of course, the tremendous progresses through the Genome Project, and understanding how disease biologies work and how they can be modulated. The evidence for this has been basically correlating the growth in genome-wide association studies with how that has impacted the progress of assets through the R&D value chain. And there's evidence that shows that this availability of better information is enabling more success. And the second is just the better use of information. Now, of course, pharmaceutical companies have been investing a lot into improving the decision-making using this new data that's available. And so that has been another driver of it. And then finally, the authors observe changes to the approval threshold. Of course, as you're developing more targeted therapies for rare diseases with high unmet need, it has a different bar for approval threshold. So where can AI play a role? Really on both of these first two aspects around how can we glean better information and how can we better use this information that we have so as to develop new drugs and therapeutic interventions for patients. And in fact, as we look out into the present and to the future, I believe we're at the cusp of an AI revolution in pharma R&D. Three reasons for this. First, the explosion of data itself. So it's expected that over the course of the lifetime of an individual, they will generate about 1,000,000 gigabytes of healthcare data. 1,000,000 gigabytes, which equates to a petabyte of healthcare data for every individual. So when you look at the size of human population, that is the promise of hundreds of zettabytes of data that will become available, of healthcare data that will become available to us. Right? This data ranges from data about genome sequences through next generation sequencing tests to map out an entire genome cost less than $100. This includes data that is getting digitized with electronic health records, medical imaging and radiomics data, wearable devices data, applications that we use in digital therapeutics, and all of the streaming data that's becoming available to us. So that's exciting that there's so much data, because data is the fuel that powers the promise of AI. But tied to this data are challenges. Much of this data is unstructured. Much of it is unlabeled. It's not all going to be integrated and mastered. And so the emphasis on the verification of this data so that it can power the next generation of learnings and insights is critical. And so we'll talk a little bit about that in the next few minutes. The second reason why we're excited about this revolution in AI is because of programmable biology. Now, over the past decade, about 20 cell and gene therapies have been approved here in the United States, and it's expected that this number will explode. And really, what is leading to the rise of all of these cell and gene therapies is the ability to really program molecular data, right? And in order to program this molecular data, we do have to develop a set of skills beyond traditional molecular science to couple that molecular science with data science, because this is ultra high dimensional data, right? So there's an opportunity here, but there's also a challenge of bringing together these schools of learning that have typically resided separately. And then finally, let's spend a little bit of minutes talking about productivity. We talked about productivity on the previous page, but I want to talk about productivity in the clinical development setting. There is a huge avenue for improvement, a huge opportunity for growth. When you think about clinical trial design in particular, right? Protocols have been becoming more complex. Over the past decade, the number of procedures that go into a clinical trial protocol have gone up by 70%. The number of endpoints in clinical trials especially in phase 3 clinical trials, have gone up by 86%. And on average, a clinical trial protocol has to be amended about 5 times through the clinical trial process. And remember, this is a process that takes several years. The overall time to go from invention to approval for an asset is about 10 to 12 years. And clinical trials, that process, about 65% of the time and cost investment is in the clinical trial design and operations process, right? So there is rooms for improving the design, and from an operations standpoint as well. Identifying better clinical trial sites, identifying investigators for clinical trials also provides room for improvement and growth. Today, there's a study that notes about 50% of oncology clinical trial sites that have been onboarded never actually enroll a patient. 66% of sites do not meet their enrollment targets. And today, only about 3% of all eligible investigators are being targeted for clinical trials. So lots of opportunity for improvement, not to mention doing so in a manner that we are addressing concerns around health equity and inclusion of various populations, where AI can also play a role in doing that in a more equitable fashion. Okay, so lots of reasons for excitement, opportunities as well as challenges, and we'll talk through what are the implications specifically for us. So with all that being said, the big focus areas where AI can help R&D are specifically, I will put them into 4 buckets. Okay, breakthrough science is the first area where AI can help in R&D. If you think about disease biology today, there's still a lot to be learned about the human body, right? And a lot of that learning is available in biomedical literature. So how can we leverage this information that today sits in biomedical literature and tie data analytics with knowledge analytics to improve our understanding of diseases, targets, mechanisms, and eventually drug candidates? So that's a big focus area. Second big focus area is precision medicine. Really at the core of it, it's about how can we identify the right patients for every treatment that we are developing and bringing to market. And secondly, for every patient, how can we identify the right treatment? How can we target the treatment that's right for an individual patient? With this availability of data that we talked about, the promise is there to tackle this important problem around precision medicine. The third big problem from an R&D context is how do we improve the probability of trial and regulatory success in clinical trials? And here, really the opportunity for us is how can we simulate in silico the likely outcomes from a clinical trial, especially as more and more data becomes available as we advance through the clinical trial process. And finally, as we've talked about, how can we use all of this data and the power that we have available, whether it's real world data, whether it's the development of in silico methods and automation to accelerate the entire discovery and development process that's otherwise costing $1 to $2 billion and taking 10 to 12 years, and really shorten that so that we can bring better medications faster and more cost effectively to patients. So with that, I'd like to spend our time talking specifically about a few case studies that help to bring to life how AI has in fact been deployed in pharma R&D and some of the learnings from them. The first case study I would like to talk to you about is around a condition called NASH. So maybe a little bit of context here. There is a condition called non-alcoholic fatty liver disease. It is in fact the most common chronic liver condition in the United States. In fact, it is believed about 25% of all adults suffer from non-alcoholic fatty liver disease. Now, non-alcoholic fatty liver disease over the course of decades can progress into non-alcoholic steatohepatitis, which is again a liver condition where the condition of the liver worsens, to put it simply, and if not treated, can lead to really, really bad outcomes, right? Outcomes such as liver cirrhosis, outcomes such as end-stage liver disease, outcomes such as hepatocellular carcinoma, right? So it's important that we try to solve for this issue of patients having NASH. The challenge is that today there is no medication for reversing the course of progression of patients on NASH. However, there are treatments that are being developed through exactly those targeted therapies we've talked about that can slow the progression of NASH and prevent those undesired outcomes. Now, in order to do that, we have to be able to diagnose whether an individual actually has NASH. And that diagnosis process today involves a procedure known as a liver biopsy, where a part of your liver, a piece of your liver is basically taken from you through a biopsy procedure. This is an invasive procedure, and it has a lot of associated risks, including mortality risks. So because of the invasive nature of this procedure, this condition generally is undiagnosed, coupled with the fact that the progression of this disease can take a long time. And so there's an opportunity or an unmet need specifically around coming up with non-invasive diagnostic procedures for NASH. So this was a problem that, in fact, we worked on with some of our collaborators. And we sought to see, can we predict whether or not a patient is likely to get diagnosed with NASH based purely upon EMR data without the need for a patient biopsy. So the approach that we took here was one where we leveraged the extant literature to understand risk factors for NASH and inform the feature engineering that we do in AI and machine learning modeling We built a supervised machine learning model using the NASH Clinical Research Network data from NIH. We then subsequently, we built a model. We got really encouraging, promising results in terms of quality of accuracy using this NASH CRN data. But we needed to validate it even further in the real world context. So we took this machine learning model and in fact validated it on real world Optum data, Optum EMR data. And in fact, the models continue to demonstrate really, really promising results. Today, we have a model that's really high performing, greater than 80% area under the curve, sensitivity and precision. This research, this AI model has in fact been validated by experts through publications and conferences. And this validation today using real world data enables its use at least in clinical trial planning, right? The enrollment of patients, site selection, number of other decisions. As we continue to enhance the validation process, including its use in clinical care settings, we envision the possibility of algorithms such as this actually being deployed in patient care in clinical settings to aid in decision support for physicians. So really an amazing application of AI and ML to come up with this non-invasive approach to predict NASH. I think a few key meta learnings. First of all, to do this, we had to take a multidisciplinary approach, bring together a team comprised of epidemiologists, experts in real world data, and data scientists, all working together towards this problem, experts in HEOR, all working together to solve this important problem. The second meta learning was the importance and rigor at the time that it takes to go through a clinical validation process. This entire process has been a 2 or 3 year journey for us. This process takes time. Developing validated machine learning models takes time in the context of pharma R&D. So that's another key learning that I want to talk about, and hopefully we'll reflect on it at the end of our presentation. I want to talk about a second case study building on the work I was talking about from a NASH standpoint. And that is specifically around taking a semantic approach to identify combination CAR-T modalities. So what's the problem here? The problem here is that, as we talked about, CAR-T is an exciting new therapy that's being used to treat otherwise several life-threatening conditions. But one of the challenges with CAR-T therapies is that they can have other immune resistance challenges as well. So, and a big area of focus in the research is how can we investigate combinations of CAR-T cells with other treatment modalities so that we can overcome this immune resistance, potentially enhance therapeutic efficacy in conditions like solid oncology tumors, and to modulate pathways during the T cell activation process, right? And so there's a lot of research happening in this space. It's exciting, but it's also rapidly expanding the research corpus, and it's increasingly becoming challenging for researchers to stay on top of all of this literature that's coming out, right? So the question that was posed to us is, can we rank and prioritize these research publications that are coming out so that researchers can develop a deeper understanding faster for these combination therapies and relevance for development of these targeted combinations? And so for that, again, we took an approach leveraging the concept of knowledge graphs and natural language processing to develop a model that could look at biomedical abstracts and the literature, identify whether or not that abstract was talking about a combination therapy related to CAR-T, and extract what those combination therapies were, the concepts that they were related to around modalities, the mechanisms of action that were being referenced, the gene modulations that were being referenced, the gene engineering that was being referenced. So we extracted all of these different concepts from the literature And we ranked these abstracts based on their provenance, whether they were from clinical, in which case, of course, it's a high rank or a high prioritization. If they came from animal models, a medium, or they was deemed of sort of medium importance. And if they came from cell lines, then of course, low importance. And through the development of this model, we were actually able to come up with this prioritization of abstracts. We were able to validate it with experts who reviewed it to ensure that the model was, in fact, enabling better prioritization or decreasing the time that was spent by these researchers to identify these relevant papers for them to look into further. In addition, we were actually improving and codifying this knowledge around combination therapies into this sort of knowledge layer, if you will, so it could be reused for other scientific questions, right? So this is another really interesting use of AI and machine learning to, in fact, codify knowledge in a way that it can be reused for this knowledge analytics process to go along with the data analytics process that is so core to drug discovery and development. And finally, I'd like to talk to you about a third application around automating the data harmonization and standardization of clinical trial data. Now, clinical trial data is recorded in many different formats and structures across studies. And when you think about the need to conform this data, for example, to the SDTM format, which is needed for regulatory submissions, this is a fairly tedious, expensive process done manually with dependence on SMEs and coders, right? And so we said, well, if I take this problem of data conformance and standardization, which is done manually by experts, how can we use AI to make that process much more efficient? And in fact, we developed a supervised ML system to map and recommend transformations that should be applied to the data so as to support the data conformance. We built a human in the loop process so that we could take users' feedback on the recommendations that the algorithm was making. And through active learning, we were actually able to even improve the future recommendations that this algorithm would make. And in fact, in the process of developing the source to target mapping using machine learning, we were able to develop frameworks that actually scale across domains and functional areas. Through this, we were able to achieve 85% faster study conformance time, going down from about 1 to 2 weeks down to just a couple of days. We were able to achieve quite high accuracy, even with a limited training data set, about 80% accuracy with a small training data set of historical data transformations and harmonization that had been done. And in fact, with active learning, we can improve accuracy up to 90%. And human in the loop ensures that all of this is still scalable in a compliant way, which is a really important part of the process, while also generating significant cost savings. In the very first year, over $1 million of cost saving, and that savings will scale further. So in summary, I'd like to wrap up my talk by talking about learnings or considerations to this promise of unlocking AI productivity in R&D. So the first is really taking a multidisciplinary team approach to problems in R&D AI. It not just involves technical experts like data scientists and machine learning engineers and data engineers, but also deep domain experts like molecular scientists, biostatisticians, bioinformaticians, all working together in a multidisciplinary collaborative way to solve the kinds of problems we've talked about. Second is the observation that the paradigm for R&D AI is different. AI needs to be grounded in the context of R&D in scientific knowledge. We need to think equally about inference, not just about accuracy. And there's a long gestation period for concept validation. All of this needs to factor into how we plan to make investments in AI in the context of pharma R&D. The third is taking a portfolio approach with the end in mind. Specifically, building a balanced portfolio of high-risk moonshots that can have high impact, but also shorter-term gains so that we can continue to make incremental progress as well as disruptive progress through AI. And finally, think about AI as a platform technology in the context of R&D. How do we build reusable capabilities around intelligent knowledge management, intelligent data management, building the right knowledge architecture, doing patient computational phenotyping at scale? In order to do these, we can build AI modules that are reusable across several functions, whether it's biomedical research, preclinical, clinical, evidence generation. And so taking that platform technology mindset to AI will be critical to scaling its impact. With that, I thank you for your attention today, and I look forward to connecting with you over the course of our conference.